Hello and welcome everyone to another episode of our DNA University's Genomics in Practice webinar. My name is Dr. Laura Varden. I'm Dean of DNA University and also an in-house clinician here at the DNA Company. And we have a special treat tonight. We have our own Associate Dean, Dr. Lachlan Crawford, who is going to be presenting a case study of a grad student who struggled with brain fog, anxiety, insomnia, fatigue, all of these issues. I mean, with the hard enough job of working to get her PhD, and how Dr. Crawford used her DNA and testing and other types of tests, which she'll describe, on figuring out the root cause of her issues and how she was able to help her. So welcome, Dr. Crawford. How are you this evening?
Thanks, Dr. Burden. I'm great. Yeah, it's good to see you. I'm always looking forward to these webinars. So I think it'll be a nice case to go through today that really ties in a lot of pieces. So yeah, I'm looking forward to it. How are you doing tonight?
I'm doing wonderfully and getting ready for the holidays next week,
Right.
being Thanksgiving for our US people. And then Christmas is right on the tails of that. So, yeah, actually, speaking of the holidays, I did want to mention that we have some wonderful sales going on for people to be aware of, for you practitioners out there that you can turn around and offer your clients. And so we've got a Black Friday Cyber Monday deal, breaking that down, and Vanny can put this information in the chat that we have for Black Friday, we have our Black Label personalized supplements will be 10% off three or more of our Black Label supplements. Actually, these are our the formulations that you'll find on our website. Also for Cyber Monday special, we have 20% off our DNA 360 plus a clinician navigation call bundle. And finally, we also are offering 10% off all of our genetic testing. And please note that the discounts apply at checkout. So I just wanted to give a shout out for that so you can take a look. And, it ends up being a great holiday gift. Also,
Mhm.
please check out our DNA Talks Digest, our issue 6 that came out October 1st, because that includes our holiday gift guide, not only talking about the products, the tests that we offer, but also other wonderful products from affiliates that we do work with, that we use ourselves. So please check that out.
That's a great idea, a DNA gift for somebody, helping them figure out their health and just learning about themselves. I think that's really awesome.
So why don't we start? Oh, I see a question real quick. Where do we find that? So Anne was asking, where do we find that? What you can do if you are asking about the DNA Talks Digest, you can go right to our website on the DNAcompany.com. You can go over to resources and there's a drop down and there will be our DNA Talks Digest. It's also available on our app, the DNA Way. Everything right there at your fingertips, including your reports, or as a practitioner, even your clients' reports. So you can actually dive into that. You can look and sign up for upcoming webinars, watch previous webinars, watch our podcasts, as well as our DNA Talks Digest. So that's all right there. And of course, it's also available in the student area, links to these things on the platform if you are a student with the DNA University. Great question.
Nice job. Yeah, always keeping us organized there. Thank you so much, Dr. Burden. Well, that sounds like a great place to kind of get started with our case. So I'm sure you'll keep an eye on the Q&A and the questions as they come rolling in. If anything needs to be answered kind of in the moment, great. Otherwise, we'll save some time at the end for the questions. So we can go ahead and throw the slides up for this case presentation that I've prepared. Thank you so much. So I want to just start. So we're talking about functional genomics case study on cognitive fatigue and focus dysregulation. So Dr. Varden already kind of introduced the idea, but um, going to the next slide, I'd like to just start by introducing these two principles for this case. So something that I work partly within my outpatient practice is going to be metabolic psychiatry. So this is the idea that mental health symptoms aren't just something that appear, but they're actually downstream effects of metabolism and actually the way that our neurons use energy. It has a lot to do with mitochondrial health. It has a lot to do with blood sugar dysregulation, as we'll see. And so this is an idea that I kind of want to share with everybody because there's new wonderful emerging evidence that mental health, which is my clinical specialty, is really impacted by many systemic things that are happening for us. So that's the idea that metabolic psychiatry looks at, and then we're looking at metabolic psychiatry genomics, right? So we're using genomics as a layer within this to say how can we use genomics testing to actually help us figure this out, how to figure out this piece of how do the systemic pieces kind of interlace with the metabolic, or excuse me, the psychiatric symptoms that kind of manifest and bring us to the doctor's office. So we're gonna be introducing maybe some kind of new ideas, but yeah, so those are just kind of two pieces I wanted to eliminate. So if we dive into the next slide, let me just pull up these notes here. Okay, so I'd like you to meet Sarah. So this was a patient who came to me. She was 29 at the time. She's a PhD student, brilliant, capable, totally motivated, but just really struggling with the day-to-day work. And not just, oh, it's taking me a long time to do my dissertation. It's hard. It was like halting, feeling like so much brain fog in the middle of the day that she had to lie down and nap every single day. She felt like she just couldn't focus. She'd get up and maybe do a bit of work in the morning, but then, she would have these large gaps in the afternoon where she just couldn't work. And then in the evening, maybe she'd have a bit of a second wind and could do a little bit more work, but she just felt so stalled out. So her main symptoms were brain fog, fatigue, anxiety was a big part of it that she started to realize. She had some sleep latency insomnia, so it would take her like up to 90 minutes to fall asleep sometimes. She was a real clock watcher around that time, so she knew it was taking her a long time to fall asleep. And feeling just kind of overall cognitive dysfunction, like word recall, and just feeling like she wasn't sharp. So as much as, maybe we're not all PhD students, but this could apply to many of us, right? The afternoon slump, the feeling like, oh gosh, I just can't quite write like I used to, or I feel like I'm just not quite as sharp. Kind of as we get older or we move into a new phase of life. So I want you to think about your patients and think about yourself in terms of like how this kind of picture could be applicable to a lot of different people. Um, One point about her is that she'd been previously diagnosed by her GP with bipolar disorder 2. So she didn't meet the criteria for full manic episodes, but she had what the GP considered to be hypomanic episodes. So the Sarah said that never really felt accurate to her. She kind of questioned it. She said, "Yeah, I struggle with kind of low mood because I'm bummed and I can't work on this dissertation. I can't seem to move forward and I have all this anxiety." And I feel stuck, but I have periods of maybe feeling a little bit better and then I get a lot of work done. So that was kind of characterized as bipolar 2, but Sarah wasn't really convinced. Um, and I think that's actually it's an important piece because misdiagnosis is kind of common when we're looking at psychiatric symptoms and not really thinking about what the root cause could be. So I was happy that we were able to kind of go deeper with her. Another piece that gave me a little bit of insight into this could be a metabolic element here is that she was skipping meals regularly. So every 3 days, she was kind of skipping lunch and she was eating a diet high in convenience foods. Now, I never want to shame foods. I also work with folks with eating disorders. So I don't want to say that, these foods are bad, and to kind of demonize things. But there are patterns of metabolism, especially if you're a sensitive person, that can pop up if we're leaning on a certain type of diet all the time. And that's what she was doing. She had a diet high in convenience foods and ready to go wraps and sandwiches and things like that. And then she'd eat one large meal with her family at the end of the day, but it was kind of late in the day. Um, So that's kind of the case there. I'm just reading my notes here. So that was her. She came to me and she said, I just want to finish my dissertation and I just want to feel like I can get through the day and feel like I have my brain back, but I just don't have the brain energy halfway through the day. So I said, let's take a look. So we did some labs. We can head to the next slide there.
I have a quick
Um,
question, Dr.
Go ahead.
Crawford.
Yeah,
Um, was she, now you said her family, did she have children? Was she married? What was the dynamics of her family structure?
great question. So she was single, and living with her family of origin. So she was living with her parents, and I think she had two younger siblings living in the house. So she was kind of finishing her dissertation and doing that grad school in the nest of her home. So she, didn't necessarily have the direct responsibilities of childcare, but she was still definitely a part of a tight-knit family group there. Yeah, but good question, right? We got to understand kind of what's going on for the day-to-day
I
experience of.
remember when I was a grad student, I had
Yeah,
my kids and everything else. So
yeah,
yeah, the dynamics are definitely very interesting, especially because a lot of the other grad students, ones that I mentored or worked with, a lot of them were married and had young children. I mean, granted, mine were a little bit older at the time, but still, I mean, it's getting your PhD is hard work. It is very intensive between the coursework, lab work, TAing, or whatever other responsibilities that you have. So I definitely feel for her. And, but I mean, just for our audience to kind of put into place to really sit in her shoes to go, okay, this is my life. So I
right.
appreciate you
Yeah, thank you for that. I
sharing
mean,
that.
it was her whole life. She was really putting kind of everything else on hold to just get this done. And can you imagine the consternation when she can't make progress, right? She's just so frustrated and she feels like she wants to. She's got the time, she's got the dedication, but she just is sitting, she'll sit at her computer and just feel like blank, just like her brain doesn't show up to the computer with her, right?
Oh yeah,
Yeah.
there.
Yeah, tough experience. And I felt for her. So this really real experience of brain fog was just super getting in her way. So we said, okay, let's do some blood work. So we really looked at a very simple panel. So we did a comprehensive metabolic panel and we did lipids. So it was very simple. You'll see that some of the stuff we talk about, we didn't even initially test for. Um, but anyway, so let's dive into the comprehensive metabolic panel. So fasting glucose. So I started to get an idea of okay, so she's got this brain fog, she's got this anxiety. Maybe let's take a look at blood glucose and see if there's something that could be happening. So we saw fasting glucose 98 for American metrics there. So it's upper normal, up to 100 is considered normal. Some ranges say up to 90, but that's upper normal. So for fasting glucose, I consider that kind of high. In terms of standard labs, it's upper normal, but I consider that high. That means that the glucose is just going to hang out in her blood. And when that happens, it hangs out in your systemic blood and it also goes into the brain, right? And so we can have high glucose in kind of the cerebral blood flow and then the systemic blood flow. And when that um, blood with a lot of sugar molecules in it hangs out for long periods of time, we start to get glycated proteins in the blood. So that's what HbA1c is. It's glycating an element of the red blood cell. And that's how we measure kind of long-term measures of what's your blood glucose like in the long run. It's kind of like a 3-month pick snapshot of what your blood glucose has been doing. And sometimes some of the other proteins that get glycated, those turn into inflammatory proteins. So that's just, I'm illustrating a little bit of the risk of like why blood glucose over long periods of time isn't necessarily helpful to the brain.
Did you
So
happen to know what her A1C was? The
we
hemoglobin?
didn't do it. I
Oh,
thought about, I could have put that in for sure. It would have been a great um, element to kind of follow up to see, okay, what was it previously and what was it after, especially if you can do at least 3 months, 3 to 4 to 5 to 6 month intervention. But we didn't do it for this case. So
gotcha.
we
I'm
just,
really glad to see you did fasting insulin because honestly, when it comes to blood sugar regulation, insulin sensitivity, just a fasting glucose is not enough to really give you that true snapshot of the way that their body is regulating blood sugar. You
Right.
really need to have that fasting insulin. So I was really happy to see that you had this in here. So I'll let you continue.
Thanks. Yeah, no, you're right. I mean, I'm so glad that I did fasting insulin. A bit funny that we didn't end up doing HbA1c, but having the fasting glucose and the fasting insulin means that you can calculate something called the HOMA-IR, which is the third point there. And it's basically just a calculation that you can put in that divides one over the other and then times it by a certain number. But it's a standard calculation that tells you if your HOMA-IR is over 2, that is an indication of early insulin insensitivity, insulin resistance. And as the same thing as fasting insulin of 11, um, that's going to indicate early insulin resistance as well. The standard labs will say that fasting insulin up to 20 or 25 is normal, but the functional levels, they are much lower. So I've seen 10 in some populations is the cutoff for fasting insulin indicating any kind of insulin resistance. There's a recent study I read actually in 2024 in Korean men that showed that 10 is the cutoff for metabolic syndrome. It's not been studied in every ethnicity, but it's interesting to say that, okay, we're starting to get some information that says 10 is actually a much better cutoff than 20 or 25. Have you seen that, Dr. Varden? Are you seeing standard labs start to change their fasting insulin
Oh,
standards?
actually, yes. I mean, granted, when I have a client come to me and, they'll usually bring CBC with differential, their CMP, maybe lipid profile, possibly if they've had thyroid issues, they'll have a thyroid panel. But I am finding, that, oh, good, they have their vitamin D, levels. Oh, hey, you've got a better profile for cardiometabolic, where they're adding in LP little a or the LDL patterns and sizes, and really starting to get into some of those other markers that are more informative. And I have seen, although not as often, the fasting insulin. I am seeing more of the A1C, even in fairly healthy people, because they're knowing that, okay, fasting glucose in and of itself can be very dynamic. That's a snapshot for that day.
Mhm.
And really, if, like you were saying, if you want a better indicator that gives you that average over the last 3 months, the hemoglobin A1C is definitely much better. But even better is having the insulin, the fasting insulin as well, giving you a much richer picture. And so I am seeing that these markers, come in. And then of course, if they go, oh, what blood work should I do? Especially if there is an issue or if they're saying certain symptoms that they are complaining of or goals that they have, I want to look to see what are we working with? What is our baseline at this moment? So that way we know when we are implementing recommendations, when we are working through a protocol, is it working? That's the test, don't guess. Let's track it. So
Yep.
that way we know what is working, what's not, and where we may need to adjust. Um, by the way, I wanted to ask you if you could explain why you have the question marks at some of the end of some of the markers, the values. I'm sure some people are wondering, why is that there?
On this slide?
Yes.
Oh, that's funny. I actually don't know. That might have been a link. I thought you were talking about in future slides. I have question marks because I'm going to leave some information for us
Oh,
to discuss with everybody
no,
here,
because
but
yeah, because
I'm not
on
I'm not sure.
yeah, because it has like the little question mark box. I didn't know if that was meant for something.
No, sorry about that. It could just be a
Okay.
formatting thing that I didn't catch before the end. It could have been some wingding like conversion that,
Okay.
appropriate.
Just
But
wanted
thank
to
you
make
for
sure,
pointing that out.
just for clarity, so people aren't going, "What's that
Yeah,
for?"
you're
So
right.
thank you for that.
Yeah, no problem. Thank you. Okay, so that makes total sense in terms of, we're tracking these markers. These are really good markers. HbA1c would be a thing that I would add. AST, ALT, we're looking at liver function. And we had mild elevation there, which just shows that we had a potential early indication of hepatic steatosis. So just the march towards non-alcoholic fatty liver disease, which can show up with insulin resistance. So those kind of can show up together. And so I'm looking at not just are the biomarkers of the immediate blood sugar regulation happening, but are some of the downstream sequelae happening, i.e. things like fatty liver disease. I was happy to see that electrolytes, renal, and other function was all normal. So that was a good thing that we didn't have to go too deep into that. The second page of labs, we went into some of the lipids here. And I mean, I think lipids are really important for mental health. You know, it's not just something that we hand off to the cardiologist, but it's something that I think mental health practitioners also need to be able to look at. Triglycerides were a little elevated. Her HDL was a little low. Her LDL was a little borderline, and her total cholesterol over HDL ratio was 4.8. Where do you like to see total cholesterol over HDL ratio, Dr. Burden?
Well, I mean, there are certain patterns that I do like to see where total cholesterol, you've got that being triglycerides being like, half of that, and then, half of that. So it's more like the patterning.
I agree.
Um, but
Yeah.
I'll tell you, I mean, when it comes to cholesterol, it's like now they're saying anything over 200 and they flag it and try to push statins when, 20 years ago or so, it actually was at 300 and they've dropped it down. And it's just, you have to look at the full picture, not just necessarily at one or two markers. You
Right.
also need to look at what symptoms is that person having? What are they complaining of?
Mhm.
Because one thing I get a lot is people coming into me and saying, "Oh, my doctor says that my labs look fine, but I feel like crap." you know,
Yeah.
or that, hey, my labs, they're saying, oh, this is out of range and they want to put me on medication, but I feel great. So you get this dichotomy, of almost extremes. And this is where we have to dig a little bit deeper and figure out, is this truly a problem? And that's what's looking at, all of these different lab markers that I had mentioned earlier, not just LDL, not just cholesterol and some of these typical lipid profiles, but actually going into more of a cardio IQ, digging in a little bit more to see that fuller picture.
Right. Yeah, I love that. I mean, it's not as simple. I mean, I threw you that saying, where do you like that? But it's so true. I agree with you absolutely that it's about the pattern. And I think more than anything here, I was kind of looking at the triglycerides because we know that that's what excess glucose and fructose are kind of packaged into. And that's what can contribute to the non-alcoholic fatty liver disease. So it's kind of noting that, okay, that part is elevated. Let's keep an eye on the rest of your lipids. And that was where we were starting from. So those are the labs. It's like bare bones. We did very little, and we started from there. And I think we can dive into what we did. So we did a DNA 360 as well. So okay, so we got some baseline labs. We did DNA 360. So these were, I'm only really highlighting three categories of things to illustrate a picture of how I went from thinking maybe there's something metabolic going on to figuring out there's very likely something metabolic going on as evidenced by the labs, and then what that metabolic thing is what is happening, and then what's the root cause of that metabolic, and how do we kind of get in there as an intervention. So I left that question mark there. I thought that's what you were talking about. I left that in because I actually want to quiz the audience and see what they think the TCF7L2 would be. But let's just start by layering in these genomics. So these three genes, the FTO, the MC4R, and the TCF7L2, collectively they tell an important metabolic story. FTO is considered the fat mass and obesity risk gene. So it is a gene that dictates response to things like satiety. It influences ghrelin, which is the hormone that makes us feel hungry. And it dictates mitochondrial energy use and adipogenesis, which is the generation of new lipid cells or fat cells. And it's going to have a lot to do with how quickly you feel full when you eat a meal. And so with this, I'm going to look at this. So A is the at-risk gene for having more of these symptoms, and T is more like the wild type. So I believe that she had the AA, so she had a double at-risk there. Now I want to pause here and not say that that's not a guaranteed sentence. I know there are plenty of folks that have FTO, AA homozygous, and they know it, and they implement lifestyle factors and diet factors that can be very protective for this. That was just something that she had, and we wanted to start off, our understanding of the metabolism in terms of like, okay, that might be impacting how her body is going to actually use energy and how we're going to store fat, and then how we're going to actually have an experience of the signals that tell us whether we're full or whether we're hungry. With that, and Dr. Varden, jump in anytime you wanna add pieces to this, because I know you're a wealth of information here, but we're already a half hour in, so I really want to get through these genes. So the second one, MC4R, this encodes the membrane receptor for cells for somatic growth and energy metabolism. For this one, the C is suboptimal and T is optimal. And so FTO I started with, that's more about satiety, but MC4R is more about how quickly you're going to be hungry again in between meals. So this one is often kind of called like the snacking gene. And when we have a suboptimal MC4R, we're going to get, again, additionally dysregulated hunger cues. And sometimes they come on too early. And then the C is also going to mean that potentially that we have lower baseline resting energy expenditure. So putting those two together, we've got FTO and MC4R really kind of painting a picture that we may have impaired satiety, we may have difficulty in regulating hunger cues, and then having a tendency to kind of snack and hold on to adipocytes and fat cells when we've kind of made them. So those two together give us an idea of how her body is kind of handling and managing energy.
Actually, we have a question that I wanted to throw out that I think would be very appropriate right now. Janice was asking, did she have any issues with her weight?
She did. So something I forgot to mention in that is that, so kind of like a side complaint or point that she was bringing up was like that she'd been gaining weight without any kind of change in energy expenditure or diet. And so she was 29 and she'd been gaining a little bit of weight. I don't think she put a number on it, but she's like, I don't feel like I have stabilized my weight. I feel like I'm continuing to slowly gain weight over the last 2-3 years. She wasn't on the scale of BMI, she wasn't in the obese category, but she felt like she was carrying some extra weight and didn't really understand where it was coming from. But that's a great question. And that's the kind of thing that you might expect, right? So I love that that question was there. Now I want to ask folks, so I want to talk about TCF7L2. This is the gene transcription factor 7 like 2. And this produces a protein in the blood, which has to do with blood glucose homeostasis. It's strongly associated with dysregulation when we have blood glucose dysregulation, when we have the suboptimal, which is the T allele. The G is the optimal allele, and we've got T being suboptimal. So I put a question mark here. Let me just describe what the gene does, and then I want folks to kind of guess at what they might think would be showing up for her. So with TCF7L2 T alleles, 1 or 2, we're going to have impaired insulin secretion, and we're going to have impaired glucose tolerance. So that fasting glucose is going to stay higher. And we're going to have impaired glucose metabolism. So on the back end, where we're actually using the glucose in the cells, we're going to have a harder time actually using it. And so that those glucose molecules are going to be packaged up into triglycerides and sit in the liver instead of being used by cells. So that's kind of a little bit of the picture that we were starting to see. So with that, I want to ask folks what we think might be showing up. TCF7L2 will be dysregulated. Yes, Janice. So it could be TT, it could be GT. Her results were actually GT. I think it's impossible to say exactly whether it would be GT or TT just from hearing the symptoms. But if it were TT, it would have been maybe a little bit more exaggerated insulin resistance. Maybe it would have been a little bit higher. Maybe her blood glucose would have been a bit higher. Um, but oh, nice. Thank you. Great. Thanks for filling that in. So she was actually GT for the 7F, TCF7L2. Very cool. Good job, Janice. All right. So that's what we got. So for the next slide, we can kind of put this all together. So FTO, MC4R, and the TCF7L2, this is going to mean that she's going to have an exaggerated insulin response to refined carbohydrates. So when she eats a meal, her blood glucose is going to go up, and the insulin is going to be released, and that's going to be an exaggerated response. And what that means is that when our blood glucose goes up and our insulin goes up and it's an exaggerated insulin, it's actually going to give us what's called reactive hypoglycemia. So it's going to release a ton of insulin all at once to really shove down that glucose as fast as possible, but it shoves it down too much, too fast, right? So we're going to get actually the blood glucose kind of crashing below 70, which is what's going to feel comfortable to the human body for most folks. And we're actually going to have blood glucose fall below that. And when that happens with reactive hypoglycemia, about 2 hours after a meal, and it can be anywhere between 2 and 5 hours, but for most people having alimentary, so food-related reactive hypoglycemia, they're going to have about 2 to 2.5 hours after a meal, they're going to start to feel really low blood sugar. And whatever that shows up for different people, for Sarah, it was anxiety. She just started to feel really hot and sweaty and anxious. Just like this dread coming on of like, oh my gosh, I have to get this PhD done. And she would just fall into this pit. And then all this anxiety would be another block for why she couldn't kind of get the work done. So exaggerated insulin response to refined carbohydrates. Her satiety signaling is reduced. She's not going to feel full necessarily in a normal way, especially under stress. Because another thing that happens with reactive hypoglycemia is that when your blood glucose goes up, you release a lot of insulin and your blood glucose comes down and it goes below where it feels comfortable. You're going to release stress hormones and you're going to release cortisol, which is a stress hormone. And then actually some people can feel hungry like 2 hours after the meal because the body feels like I don't have enough sugar. I need fuel. Even though there's plenty of fuel that's existing in the body, it's just been stored in ways that aren't exactly accessible based on the genetics. So I'm hoping this is kind of painting a picture that's making sense. I'm happy to take questions, but generally we're getting that there's a bit of a metabolic picture that says that there is fuel in the body, but she's not having a consistent enough pattern of accessibility of that energy. There's either too much sugar or not enough sugar when her sugar drops below. Does that kind of make sense, Dr. Borden?
Oh, absolutely.
Okay,
And very nicely explained as well. Thank you.
good stuff. All right. Um, So with all that, you can expect with the MC4R gene, the snacking gene, um, we're going to get the cravings and we're going to get cravings for carbohydrates and things that when the blood glucose is going low, because that's the body's attempt to kind of come back up to normal. So she was skipping a lot of meals, but I think she was snacking in between as well. So it was just kind of dysregulated cues coming in. All right. So I mentioned that she noticed that her anxiety was really tied to after she would eat a big meal, her anxiety and her fatigue would kind of come on at the same time. And she had significant mood and cognitive swings tied to food. So there was like a lot of psychoeducation that was kind of happening and learning about patterns. We didn't do a diet recall because I don't love doing those because some people love them, some people find that they're just like really kind of too much detail, but we just kind of talked broadly about her patterns of food and then she started to kind of put things together and say, oh yeah, like when I skip meals, I don't feel great. And then when I eat these carby meals, I feel even worse. So she was kind of starting to understand. She unfortunately felt like she couldn't eat anything and feel okay, but we worked together to kind of get her there at the end. We'll get to that. Okay. So what slide are we on here? Okay. Great. So I think we can go on to the next slide. Awesome. Okay, so this is the second section of the DNA 360. And I wanted to look at, so we know that there's a metabolic piece and we know that there's a mental health piece. Those could be independent of one another. I wanted to see if those could be potentially related. And this was the slide that helped me determine that, if that could be a piece. And I was looking at basically how does her body handle oxidative stress? If you remember, I was saying when blood glucose is high for a consistent amount of time, so like 2, 3, 4, 10, 20 years of high blood glucose, we start to get these glycated products and create actually reactive oxygen species in the neurons of our brain and in lots of different parts of our body. But when we're talking about mental health symptoms, they show up and they create reactive oxygen species in the mitochondria of our neurons. And that makes it really hard for our neurons to work properly and create neurotransmitters and establish things like focus and concentration and cognitive work. So I'm kind of painting this picture. I'm using specifically talking about reactive oxygen species because that's how the metabolic piece translates into mental health. And so these genes, GPX and SOD2, oh, and FOXO3, I don't think I put SOD up here, but we'll talk about um, what SOD2 would have looked like. So GPX, this is the second part of the antioxidant pathway, SOD2 being the first part. And I was looking at this because basically it's what takes hydrogen peroxide and converts it to water and oxygen, which is the second half of what we would do to those reactive oxygen species. Once they're in the neurons, once they're in the mitochondria, they're doing all sorts of work. And we want to be able to neutralize them so that the mitochondria don't have to deal with that mess. Just kind of gums up the energy production systems in the neurons. So GPX would be the piece that kind of clears out the second half of that. For GPX, we had reduced glutathione activity here. CC version means it's a fast enzyme and hers is slowed. So she can't handle as much reactive oxygen species. So that's going to mean that her high glucose, someone who had, really fast GPX, they might clear out those glucose reactive oxygen species faster than her. So she was more sensitive to the blood glucose in her brain. Her neurons were more sensitive. So she didn't have that protection that somebody else might have if they had a GPX. If she had slow SOD2, and I didn't put her SOD2 in here, but if she also had slow SOD2, which would be CC, which is the first half of the pathway that eats up those reactive oxygen species, it would mean that both are slow, and that would mean that we had like a double whammy in terms of having a really slow antioxidant pathway there. I didn't put hers in, so we can only speculate, but the GPX gave us some information that, okay, she's got some sensitivity there. Along with that, we had FOXO3. Now, I wish we could do a little bit more of an interactive thing and just say if anybody remembers what FOXO3 is, but I'll just kind of remind folks here that this is the master control for the antioxidation profile. This is a transcription factor that switches on genes to combat cellular damage broadly. So it's like a homeostasis molecule in the body that when we have a functioning FOXO3, then we're going to have a better antioxidant protection. And if we have a suboptimal FOXO3, we're going to have less antioxidant protection. So she had a GT. And I actually wanted to talk this out with you, Dr. Barden, because, I think that the GT, the G allele is G, let me just say this. So G is increased efficiency, and that's associated with, slightly longer lifespans in certain populations. So the antioxidation support is going to help you fight off all sorts of things that lead to chronic disease. So longer life makes sense. And then the T is going to be slightly less function. Now, you'd think that GT would be deleterious, but my understanding is that GT is actually kind of in the middle, and it's actually not a terrible genotype to have. What's your take on the GT TT for FOXO3?
Well, when you have a G allele, it actually, like you were saying, enhances the ability for what we call the longevity gene, okay? Because there's a lot of research that has been done on FOXO3. It is one of the most widely studied longevity genes because it influences cellular stress response, inflammation, mitochondrial function, and obviously, therefore, healthy aging. And, when we're talking about the T allele, that is your common allele. There's nothing necessarily negative about it. It's just that you're common. So when you have a G allele, so if you have a GT presentation, or especially if you have a GG presentation, that's protective. That actually enhances that longevity response. So really the GT just means that the individual carries that one copy of the longevity associated G allele and one copy of the wild type, regular allele and the T allele. So the functionality really creates that, in a way, an intermediate phenotype, not as protective as a GG, but significantly more resilient than a TT.
Right. Okay, thank you. Yeah, someone's saying that the expression is that GT won the life lottery and GG won the mega lottery. So I kind of love that. So she's
Yeah.
right. And so she's not doing too bad with that, but nonetheless, if this were like a perfect, study case, it might have been that she had a TT just to kind of illustrate the point. But nonetheless, I still think that this profile put together showed me that she was going to need a little bit of antioxidant support. The CYP2R1, that is the enzyme that's going to help turn inactive vitamin D into active vitamin D, which is going to play a role in inflammation and energy. So that gene can go in many different categories. In fact, vitamin D itself has a direct impact on insulin as well, fasting insulin levels. So if your vitamin D is low, excuse me, if your vitamin D is low, then it's not going to be able to have a protective effect against rising fasting insulin levels. If you do have a high or a sufficient vitamin D, it's going to be a little bit protective against rising fasting insulin levels. So this is why it is metabolic. It does have to do with blood sugar, but it also definitely directly has to do with inflammation, knowing that vitamin D is anti-inflammatory and it can really help with energy production as well. So G is optimal for this gene. G is suboptimal, A is optimal. And there's a stat here that I want to share that I think is important for this, is that 53 to 70% of what you can measure in a person's serum is predictable by a person's genetics, which kind of floors me. I think it's so important to kind of know what's going on with a person's vitamin D. I didn't include it in the labs, but seeing that this showed up in the DNA 360, we certainly started to pay attention to her vitamin D, it being so important both for the insulin, as I said, but then also for the inflammation. Um, let me just poll the audience here. What gene do we think would be helpful as part of the pathway when we talk about vitamin D production? What's the other gene that would be helpful to look at for vitamin D production? Let me see if anybody is engaged in thinking about this.
And remember, if you have any questions, please put them in the Q&R or Q&A. Okay, so we've got people saying VDR, which is the vitamin D receptor, others saying vitamin D receptor, and even the GC, which is the vitamin D binding protein, that transport gene. So we're getting a lot of VDRs. So
Yep, fantastic. GC and VDR. Those are the ones that were on my mind, absolutely. CYP2R1, yep, CYP2R1, that's the one that's in the presentation there, absolutely. So those three, you can put them in a genomics pathway and kind of tell a story with that. So yeah, absolutely. Okay, so with that, just kind of coming back to this slide here, together thinking about the first slide with FTO, MC4R, TCF7L2, and this slide, I think that they create what I call like an energy fragility profile. So we've got this propensity for destabilization of energy. And then we've also got sensitivity to inflammation and reactive oxygen species. So not only do we have the dysregulation, but we are sensitive to it. So that kind of, to me, put it together and said, okay, this mental health picture could certainly be a part of, or at least caused in part by the metabolic piece that's happening. When her glucose swings, she produces more oxidative stress than average, and she has less capacity to buffer it. And that's part of what was leading to fatigue, fog, and burnout for her. So that was an interesting piece. Now, for the next slide, we can add the neurotransmitter layer. And this is where her clinical picture really becomes complete. Now, I kind of want to, I'm going to go through these. I'm not going to like describe every single one of these genes in, its full depth, because I want to like give the whole case picture. But basically, COMT, her COMT was AA. We'll just go through a few of these. So COMT is basically dopamine and norepinephrine breakdown. So these are catechol O-methyltransferase. We're breaking down catecholamines, which are essentially the activating neurotransmitters, or a subset of the activating neurotransmitters. And so if that COMT, which uses methylation, by the way, if that COMT is slow, that means that we're going to have higher levels of those activating catecholamines exist in the body for longer. So that means we're going to have a higher sensitivity to stress. So yeah, dopamine, epinephrine, and norepinephrine. A is slower. Sorry, A is slow. G is going to be faster for that one. Now, same sort of thing with MAO. This is for serotonin and dopamine and some of the other monoamine breakdown. So it's going to do the same sort of thing that COMT is doing, but for slightly different molecules. And we also had a slow MAO. So TT is the slow version, and C allele is the faster version. So with having a TT MAO and a AA COMT, she had higher levels of existing serotonin, dopamine, norepinephrine, the stress hormones, kind of activating hormones when something would happen. So if she was feeling stressed or a deadline was coming up, or she fell asleep and had a nap and slept through something that she was supposed to do, she'd have this stress grip her, and it would just last a lot longer than it would for anybody who had faster comps or faster MAOs. And she found that this would kind of happen as part of her daily routine, is that she'd get really sluggish in the afternoon and just not be able to function. She'd take a nap and wake up, excuse me, and then feel this kind of second wind of, oh my gosh, I didn't do enough today. And so she had a little bit of a burst of energy in the evening, just kind of from stress. And then she would have that meal with her family, and then she would have a really hard time going to sleep because she wasn't breaking down some of these activating chemicals. So she had kind of like a bimodal day where she had a burst of energy in the morning and a burst of energy at night, and it just wasn't working for her. The 5-HTT-LPR, this is the gene that codes for what we like to call the revolving door of serotonin in the presynaptic neuronal membrane. So basically, are we releasing serotonin properly, and are we able to reabsorb serotonin properly? This is also the target for SSRI genes. And basically, if you have a long, it's L or S, long or short copy of this. And if you have a long copy, that's going to be optimally functioning. And the short copy is going to be suboptimally functioning. So she had LS, which is not, the most difficult, but she definitely was experiencing irritability. She felt flustered when things were not happening according to plan. And she was very focused on detail. And this was part of what she was thinking was contributing to her mood swings. And she definitely knew that she had to have coping mechanisms to manage her stress, because she felt like she wasn't able to really handle stress as it came in. I think partly because of the COMT and the MAO, but also the 5-HTT-LPR was painting a picture with that too. Am I going too fast here, Dr. Varden, through this? I don't want to zip through it too quickly, but I want to give a full picture.
Well, I actually would ask our audience. So put a 1 in the chat if she's going at a speed that you are able to follow, that she's covering the information.
All right, Cindy, that's
I'm
great.
seeing one's perfect. Excellent. So
Fabulous. Okay.
fantastic.
Thanks, everybody.
I know that there has been a couple of questions about, so what do you do? What do you do with this
Yeah.
presentation? So,
Yes,
and
totally.
she
And
is
so that's
getting
why I
to
want to
that.
get there.
Yeah,
Yeah.
she is getting
Okay, perfect. So we did 5-HTLPR. So DRD2, this is the dopamine receptor density. So she had AG. This is kind of rare. She had normal density. If you have really high density, right, that's the idea that you're going to have really intense reactions to things. And if you have low density, then maybe you're going to have kind of limited reaction to things. You're going to be kind of a cool cucumber. But so it was kind of in the middle. It didn't really contribute to her picture, but I just wanted to show that, when we do these DNA 360s, sometimes genes really line up and tell a story. And then you might expect, I thought maybe she would have um, a, a high density of DRD2 because she was having stress reactions to things. But, it showed up as AG, which is considered kind of like a normal middle Zen state. The BDNF, this is the gene that codes for the production of brain-derived neurotrophic factor, which is basically what I tell patients all the time. It's like miracle grow for your neurons. It's something that helps them grow, it helps them mature, it helps them make connections, and helps them be maintained. So every time you learn a thing, every time you practice something, you're creating new neurons. And we want the ones that we're trying to support be the ones that are sustained, and we're trying to let the pathways that we don't really want to hang on to, like the fear-based pathways or the reactive-based pathways, the habits that we're trying to let go of, we want to be able to let go of those. So we want to use BDNF to kind of train our brain. And the A allele is low production of BDNF, and G is going to be higher. So this meant that she had low BDNF, low neuroplasticity, and it also meant that her brain was going to tire quickly under stress or under low glucose because it didn't have the ability to kind of make other pathways around things. When she got tired, she wasn't able to kind of rely on those new forming pathways because they just didn't have enough BDNF to kind of be supported. So it was kind of painting a similar picture. She also had a harder time recovering from oxidative stress in neurons from the glucose issues. And that's part of the BDNF too, is the neuroprotectivity or neuroplasticity and being able to function when there is neuronal damage happening from the mitochondria damage. So if we put all these together, all of these on this slide, we get, actually all three slides, we're going to get what I call a neurometabolic sensitivity pattern. We've got the metabolic stuff happening. We've got the reactive oxygen species, which is likely a contributing factor. And then we've got the neurotransmitter sensitivity that is going to show us the stress sensitivity that says, okay, why is she having so much reactivity to these pieces? All of these are going to kind of tell a story. So when her glucose drops, her dopamine is going to drop, her serotonin is going to drop, her stress hormones are going to go up, and her symptoms are going to appear just like that. So that's what was going on with her. Neurotransmitter is very sensitive to all of these pieces. So that's what we got. That's the case study. So I think the next slide, if we go on to that, we'll see what we got. All right, so treatment, this is what people want to get into. So we definitely had a lot of psychoeducation sessions about how energy regulation and metabolism is about so much more than cardiovascular risk or diabetes risk. I really had to sit down with her and say, I'm not a cardiologist, but your lipids tell me a lot, and your blood glucose tells me a lot about what's going on. And it's not hard to grasp. I mean, all of us have had a high sugar meal or something and then felt sleepy after. But I had to explain it to her to say that she had this sensitivity kind of turned up to 11 because of her genes showing that she had all this sensitivity to insulin and sugar and reactive oxygen species. So she got it when we kind of put it all together. Um, and she understood that when the body's insulin and glucose levels are inconsistent, so really up and down, it's going to send inflammation to the brain, and the brain doesn't have that resilience to kind of fight against it. Also, a thing that's important to know is that peripheral resistance, peripheral insulin resistance, meaning systemic insulin resistance in all of your organs and in your muscles and your fat, it's correlated with cerebral insulin resistance as well. So even though the neurons in your brain don't require insulin to use glucose, it still makes it harder. So if you have high insulin, that's just if you take one thing away from this talk, if you have high insulin, it's going to make it hard for your brain to function because it has a hard time using that energy. And so all of this was kind of painting a picture that the neurons were actually acting kind of like they're starving. And that's what her brain fog was about. So we had to talk about nutrition. We had to talk about metabolic regulation and energy regulation. It's not about eat this, don't eat that. It was about how do we help your brain cells actually get the nutrition that they need and the rest of your body, of course. So we started with nutrition. Um, I think something that I didn't mention actually at the beginning of the case study is that this person actually had a history of disordered eating in college. It wasn't diagnosed as anorexia or any of the DSM categories of eating disorders. It was just like vague eating disorder behaviors that were happening in college. And she said that they came on when she was the most stressed. So I really noticed that when she said that she was kind of skipping meals here or there. And so what I wanted to do with this nutrition strategy was that I did not want to say, cut out this, cut out that, cut out that. I'm really sensitive to making sure that I don't contribute to any kind of, increase in disordered eating. And so I use the crowding out strategy, meaning I want to give her all sorts of things that are going to be good to eat, that are going to be supportive to eat, I should say, and eventually, and say like, yeah, you can have that bag of chips, but eat the apple with the almond butter first and drink a glass of water. And then you can eat the chips if you still want them. And over time, it kind of started to crowd out. But just as an idea here, the nutrition strategy for metabolic intervention for this is going to be really anchoring around protein, fat, and fiber. So we said 25 to 30 grams of carbs, sorry, 25 to 30 grams of protein at each meal, 3 times a day. And even just that was an event, right? I had to get her kind of, getting used to having smoothies in the morning or something that she could do consistently every day when she wasn't used to it. We added in fiber, so we certainly got up to, we didn't go all the way to 30 or 40 grams right away. We started kind of small, but we got up to, I think it was 40 grams of fiber a day for her. And I said I wanted her to start eating antioxidant rich meals. So things with eating the rainbow, berries, leafy greens, herbs, bright anthocyanidins, bright carotenoids, things like that would be really helpful for her antioxidants. And in terms of the FTO and the MC4R, remember we talked about sensitivity to carbs. So I didn't say cut out carbs. I said change your carbs for complex carbs with a low GI index, low glycemic index, meaning that when you're going to eat them, they're going to turn into glucose slower. So anything with bran or fiber, whole grains is going to be helpful. Also eating your carbs with protein, with fat, sometimes with vinegar. So there's an idea that if you have some acetic acid, which is the constituent in vinegar, and it can actually slow down the rate at which you break down glucose, or you break down starches into glucose. It's minimal. For some people, it can be effective. So I said, just let's throw all of this at it. So let's give you proper nutrients and try and slow down how quickly we're going to break everything down into glucose. And if I can change the slope of her glucose curve from something like this, like a mountain, to something like this, like a hill, then that's great. That's what we want to be doing for her metabolic health. So we were reducing her glucose spikes without restricting foods. And I had her do electrolytes and hydration earlier in the day. So I think she was drinking Gatorade and stuff at night before she was going to bed. I had her do electrolytes earlier in the day to get that hydration in, to get her metabolism kind of going. And we were doing smoothies and savory protein-rich breakfasts. I have a note on what we did with our continuous glucose monitor, which is what we did, but I don't want to take too long on this. I want to get to questions. Let me come back here. Okay, so increased antioxidant capacity. We did do liposomal glutathione for a short while just to kind of bump up her glutathione levels. Vitamin C, NAC was really great for this, and vitamin D, of course, we knew that that was going to be needed. Improving mitochondrial health and fuel efficiency. So acetyl L-carnitine is amazing for mitochondrial health. It is a molecule that helps shuttle precursor molecules into the mitochondria. So it's kind of doing part of the work for the mitochondria. And actually, a lot of people feel their energy improve once they have acetyl L-carnitine added in. We've got to remove the antioxidants, but then actually giving the mitochondria a little bit more support in this way is helpful. Same thing with CoQ10 is used in the cellular respiration pathway or the oxidative phosphorylation pathway. So that can be a good thing. A lot of people on statins take CoQ10, which is a great thing. And then B vitamins for energy in general. And the neurotransmitter balance, we did tyrosine, which is the precursor for dopamine. And we did 5-HTP, which is the precursor for serotonin. And we did that mostly, I mean, she has the neurotransmitters, and she has slow COMT and slow MAO. So you might be surprised that we're kind of adding these in. But that was really just for kind of consistency. If she just had like a little bit twice a day, we weren't giving her 500 milligrams. I think we're giving her much less, but she just had a little bit more consistency in her neurotransmitter input. And that ended up being helpful for her mood as well. So for the next slide, we talked more about, yeah, some of the other interventions. Sleep, this I tell literally everybody this, whether your sleep is great or not. I tell everybody to get morning sunlight exposure, and really try and have a consistent bedtime and a consistent wake time. This is really important for your circadian rhythm. We didn't talk about her clock genes in this at all, but you know, part of being able to have your cortisol do the diurnal rhythm, which is helpful, and not have these big rises in cortisol at the end of the day, means that we actually need it to rise earlier in the morning. And the best signal for that is sunlight directly in the eyes. So I started to try and get her to go for a walk first thing in the morning, come home and have a smoothie, and then have a walk directly after the biggest meal of the day. Those were two kind of behavioral interventions that I put in there. I tried to get her to do the sauna. She liked it, but she didn't have a membership to a gym where she could do it consistently. But I said that for BDNF, and then also for the metabolism, so the FDO and the MC4R, we can support some of that with sauna and heat exposure. And we were working up to exercise with her. I think she was doing yoga, as I said there, a couple times a week, which was great. She was doing that from home. And we were working up with the walks towards doing some more strength-based exercise at the gym. Her
Dr. Crawford,
behavior, yes.
I just wanted to interrupt for a moment because Janice had a very important and interesting question asking about her TPH2 result because she had mentioned about a BDNF epistatic effect. But I want some clarity, or I'm going to offer some clarity on that because this particular client of yours, her BDNF was an AA presentation, a homozygous suboptimal. And the only time that there is an epistatic effect of TPH2 on BDNF is if both of those genes are GG presentations. So in this case, it wouldn't have mattered as well. And I say that loosely, but what I'm saying is it doesn't really matter what her TPH2 is as far as epistatic effect because they were not both GG. So I wanted to have some clarification on that.
That's great. Yeah, thank you for that. I mean, it is really good to note that often we'll say like, if you look at a gene in isolation, we're like, GGBDNF is the best and AA is not the best. It's easy to say that, but actually there's all of these different interactions that are important to kind of note. So thank you for pointing that out. Yeah. Um, yeah, Genesis, thank you. That's great. So behavioral repatterning, she was using the Pomodoro method. She got that from her university. I don't think that there's great research on the Pomodoro method, which has to do with like the amount of time you're working, the amount of breaks that you take. But I did say that when you're going to take a break, check in with yourself. We did metabolic mindfulness and we said, how do you feel? Do you feel tired? Do you feel hungry? Do you feel irritable? Do you feel exhausted? Can you get up and go for a 5-minute walk? Can you just get up and go upstairs? Do you need a little snack? Right? But do it from a place of when was the last time I ate, managing blood sugar, as opposed to just do you feel snacky and uncomfortable and craving. So we really did a lot of education around metabolic mindfulness. And that was the intervention. It wasn't that, elaborate. We didn't do a ton of IVs or anything really intense, but she really kind of started to get the hang of it. And when she started to feel better, she started to sleep better. She started to notice that she wasn't needing as many naps. Her anxiety started to come down, and she started to say, okay, I'm not having as much intense reactivity to a short period of time in the afternoon when I'm not able to work. I'm kind of bouncing back. And actually, even those episodes of when she wasn't able to work started to be reduced because we just weren't having those fluctuations in blood glucose as much. So I think the next slide is just a summary of some of the biomarkers. Yeah. So biomarkers, and then the last slide is just going to be kind of experiential outcomes. But her fasting glucose came down, not hugely all the way down to 70, but we got it down to 85. Fasting insulin went from 11 to 6, which is much, much better. It would be great. I mean, over time, we could bring it down even more, but that felt really good. Her HOMA-IR went below 2, which is the clinical indication that we want to see that she's got reduced insulin resistance. And then, like I said, she had a continuous glucose monitor, and we were seeing that instead of spiking and dropping twice to 3 times a day, even, she was having glucose spikes 1 to 2 times a week. And she was able to connect directly when that happened. That's when she needed to nap. So it was real incentive for her to have this kind of biofeedback to understand what was driving her symptoms. And then the last slide is just summarizing the clinical outcomes that she had improved focus, clarity, and mental sharpness, more sustained physical energy throughout the day, fewer naps, mood stability was improved. Her sleep time went from like 90 minutes to under a half hour. She was still checking the clock, but I was happy that she was moving in the right direction. She noticed that her anxiety was better. Her weight gain halted. She didn't lose weight, but it did halt. She felt stable. And she had no disordered eating relapse, no increase in behaviors, which was exactly what I was going for. I want to help her feel supported and not feel restricted or more stressed out by the intervention. And I believe that's kind of it for the case. When we stabilize how the brain is fueled, we stabilized how the mind can function. And genomics didn't just explain her symptoms, it really personalized and let me know how to approach the metabolic profile and the mental health profile. So that was Sarah. So thank you for joining me for that. And we do have almost 20 minutes for questions, which is great. I think that's more than we've had in previous webinars. So thank you everybody for going through that with me. I'm interested to know what kind of questions we
That's
got.
a great case. And I know that there are some questions, specifically about her case. And then there's some other questions. So I am going to bounce around a little bit in the order, seeing that we are fresh off of the case study. And let's see. Janice actually had a really good question because she said, how do you come up with a decision of disordered, in quotes, eating, as opposed to just a hectic schedule or emotional ups and downs?
Great question. This is something I actually just did a talk at a conference on. So I was talking to practitioners to really think about how to put an eating disorder hat on when you're screening somebody. And one thing I like to do is ask people about food rules. What are your food rules, right? So people often say like, oh, I don't eat dairy, I don't eat gluten, but people might have more detailed food rules. And then also, how much time and energy and effort does it take to maintain your food rules, right? So people who might be skipping meals or feel really regimented in what they will or won't eat, it might take up a big part of their day and actually take up a big part of their energy and their mind to be keeping up with those rules, if that kind of makes sense. So whether it's restrictive, whether it's binge eating, whether it's purging or whatever behavior it might be, it starts to take up a lot of time in the brain. So just ask a person kind of what's their relationship and how much stress is that causing. And of course, if anybody's exhibiting actual behaviors like purging that I mentioned, that's something that you want to be able to create a really honest relationship where they can be honest with you about that, and you can support them in that. But if you're talking about restriction, if it's just like, oh, it's just busy and I just happen to skip breakfast, then you do the psychoeducation and talk about, okay, we're gonna really try and eat breakfast. Like, let's try and do this. And if they're really not able to do it, then you gently can kind of dig in and say, like, what's going on here? Like, I've noticed that this is really difficult. Are you having any thoughts of wanting to, and you can do kind of like an eating disorder screening questionnaire where you're saying like, is any part of this as a way to manage the way that your body looks? Is any part of this a way to manage how you feel about your body? And those kind of questions can kind of open that up.
Wonderful, thank you. And actually, Dirk has got a great question that kind of dovetails off of that, but specifically works into a gene. And he said that, or actually, the antioxidation pathway. He says, given her ROS, reactive oxygen species, and her lifestyle, PhD workload, would carrying around a molecular hydrogen water bottle make sense to clear the ROSs? What are your thoughts about some of these biohacking techniques and other things that you've seen?
I have to be really honest. I have to declare ignorance about the molecular hydrogen waters. I tend to lean on, the evidence that's going to point me to some of the supplements and lifestyle pieces that are gonna be the most impact. But I'm never against if somebody has found something that works for them and they believe that this is helpful for them. I'm all for it. But I would have to know more about that particular intervention. Do you know any more about it that I can kind of learn about from
Well,
you?
yeah, actually, I mean, I think it very interesting. I personally haven't used it myself. We did a webinar. I did this with Justin, where we were talking about different biohacks, different, popular interventions. So I do, uh, you know, encourage you all to go and watch that. It was basically we were debunking different popular things, but sometimes it's not always debunked. There's actually some science behind it, theoretically great, and anecdotally, there have been some great things. Justin is a big proponent of it. I personally have not used it, but it doesn't mean that it could not be beneficial in some way.
Sure.
So
Yeah.
I recommend, and I believe Vanny just popped that in the chat. So thank you so much. Um, Dee had a question, and I believe she's talking about the FOXO3 gene that we talked about, because she's saying, what if the result was GG, doing like that superhuman, that really good. Now, granted,
Mm.
this
Yep.
is just one gene.
Yeah.
They don't exist in a vacuum, but real briefly, do you have any thoughts on how that may have affected or benefited her if
Yeah.
GG presentation?
Yeah, I kind of love that, right? If we paint a whole picture and we say this is what was showing up, and then what would happen if we changed this one piece, how would that shift the whole picture? It may have an impact, right? Because the FOXO3 GG, that's your kind of master antioxidant profile. And as we know, I didn't say what her SOD2 was, I don't remember, but her GPX was not the full suboptimal, but it was heterozygous. And so maybe the GG FOXO3 would be enough to help her mitochondria and her systemic cells be resilient against any kind of reactive oxygen species that would be generated. She would still have a lot to contend with because she has the high glucose and she has the high insulin, which is self-inflammatory, and the low vitamin D. So maybe she would have had, it's impossible to put a number to it, but like one of the four elements of all of those inflammatory pieces might have been a little attenuated, but certainly not all of it. So I would say that she would maybe have a little resilience. Maybe what would happen is that she wouldn't start seeing these symptoms until she was 33, 34, 35, if she had more antioxidant protection. Like I think with the insulin and the glucose dysregulation, it's all about length of time that you have that showing up and the amount of your life that you're exposed to high glucose and high insulin, um, that's going to do kind of cumulative damage. So if you have more antioxidant protection, maybe you have a little bit more protection for a little bit longer, but eventually that's going to be overwhelmed if we really have high glucose and high insulin, along with the low vitamin D, along with the GPX. So I think it would change something. I don't know that it would change everything so that she would skip through life and have zero antioxidant issues for the rest of her life, I think. Yeah.
Well, we've got Susan asking, you had mentioned about acetyl L-carnitine, and she's asking, is that better than the L-carnitine fumarate for the mitochondria? If so, what is L-carnitine fumarate best for?
I don't know. In full disclosure, I think I just use acetyl L-carnitine. L-carnitine fumarate, I'm assuming, is more of a metabolite that's going to fit into the Krebs cycle, being the fumarate piece. So I wouldn't be able to answer kind of the clinical difference there. It's something I would have to look up. Yeah, but thanks for sharing that. Do you have thoughts on that, or is it just kind of a piece that?
Um, well, I mean, when you're talking about more of that science-based comparison, um, really the short answer is that your use of the acetyl L-carnitine is superior, okay? Because it crosses the blood-brain barrier. It increases that acetyl-CoA, supporting neurotransmitters, improving that mitochondrial efficiency, and therefore having neuroprotective benefits. Where for the L-carnitine fumarate, um, for that, it really delivers the carnitine plus fumarate, which feeds into the Krebs cycle, like you were saying. So, absolutely supporting ATP production, especially this is very valuable when it comes to cardiac muscle, physical energy. So there are different uses, and you also have to look at what you are familiar with, what you're comfortable using. Do your research, take a look and see, what forms are best for what, and then if you're a practitioner, then obviously use your best judgment. So but
Yeah, thank you for that.
good question. Oh, Lance is asking, how long did the treatment take, like when you worked with her? What was the time period, the time span?
So we worked together for over a year. We started to see some of these changes. I mean, like I said, some of the meal and lifestyle changes kind of took some time to kind of take effect, but certainly she started to feel some symptom change within the first 3 months. We really got, and then she started to see a change in like, oh, I'm sleeping better. Oh, I'm needing slightly fewer naps. And over the next 3 months, she really made a big difference. So at 6 months, she really saw a peak in her symptom improvement, which might feel like a long time, but when you're working with the metabolism, you're teaching all of your systems to talk to each other differently. So we've got to be patient with this kind of a thing. There was a lot of encouragement and a lot of feedback that was really helpful for figuring out how to keep her motivated when she felt like she was, getting bored or getting, just feeling like she wasn't getting better fast enough. We stuck with it, and she was seeing improvements at 3 months. At 6 months, she was doing great.
Fantastic. So Shamila has a question. She said, so the impaired glucose response you described for the suboptimal TCF7L2 is that there's an increased secretion of insulin. A question. Is that how you were explaining?
No, no, there's not an increased secretion of insulin. You have a reduced, I'm trying to remember, I think you have insulin issues kind of on both ends. I think you're going to have a difficulty with insulin secretion, and then you're going to have a difficulty with insulin response at the end, at the cell level. I'm trying to remember if it's actually both ends, but
Well, I mean, really, when you're talking about, well, the short answer is not exactly, when you're really, looking at that, because when you have a suboptimal variant of the TCF7L2 that doesn't cause increased insulin secretion, it's a cause of impaired insulin secretion, especially in response to glucose. And this is the key reason why TCF7L2 is one of the strongest genetic predictors of type 2 diabetes. I mean, this is something that we could really break down, and we do talk about this in other webinars. I know that we don't have a lot of time left and there are other questions, but when you start looking at how TCF7L2, its role in glucose sensing, insulin secretion by those pancreatic beta cells, GLP-1 mediated insulin release, then you can start seeing the function of that and where others play in, like the SLC 3088, for the zinc, because that helps in the pancreas and insulin production, etc. So, being able to see a profile in someone's or in your client's DNA 360 in their diet nutrition report for this, that can give you insights of, what might be triggering some of the effects of blood work markers that you're seeing or issues that they are complaining of. It really, is taking all of this information and piecing it together to provide you a more personalized approach to be able to help them.
Good answer. Thank you for that. Yeah, I was seeing here it's talking about the beta islet cells of the pancreas, which are the ones that are going to produce and release that insulin that get impacted by that TCF7L2.
Yeah, she commented in the chat saying she asked the question about the insulin because it was said that the patient had an exaggerated response.
Thank you for that. Yes. Okay, great
Yeah.
clarification. So I was talking about she had an exaggerated insulin response because of the speed at which her glucose was going up. So the amount of insulin that your body releases is in direct proportion to how fast your glucose goes up after you eat a meal. So yes, she had an exaggerated insulin response in the short run to each meal, but over the long run with the TCF7L2, you're going to have difficulty with insulin. So that's kind of a short answer to say that short run versus long run, a little bit of a difference. But because she was eating really convenience meals with these carbs that were simple carbs that broke down really, really quickly without a lot of protein or fat to kind of slow that down, her glucose shot up and therefore she released a ton of insulin as a way to mitigate that. So that was her exaggerated response. But thank you for clarifying that. Yeah.
So Carl is asking, would something like red light therapy help in this case? What are your thoughts?
Sure. Yeah, absolutely. Red light therapy. I think if she could do that and saunas, I think those two could be very helpful for helping to support the metabolism and to support some of the detoxification. Yeah, absolutely.
Okay, let's see what else. We have a question, and actually, I think I'm going to dive into this one a bit. And let's see. So this one is from Mickey. And when you were talking about the cholesterol, the lipid profile, okay. So Mickey is saying that they've seen presentations where small and medium LDL particle sizes are high, even in an individual with good cholesterol levels, with total cholesterol below 200. Okay. Any thoughts on why this would happen? Also, HDL particle sizes would be within range. And Mickey's even saying that they've seen individuals with high 90s fasting insulin. Well, actually, fasting in
Blood
I
glucose.
think blood glucose, because insulin, if that was that high, that would be really bad.
Yeah.
But normal, so fasting glucose, I'd suspect that was just a mistype, and normal fasting insulin, but their hemoglobin A1C is pre-diabetic range. Okay, so I'm going to give some kind of information on that, and if there's anything that you want to comment on, but really what can happen is that in insulin signaling, when we're talking about hepatic glucose control, yeah, you can see normal fasting glucose insulin, but pre-diabetic A1C and often that small medium LDL particle size increase because, for example, key genes, TCF7L2, your SLC38 that I was just mentioning, your FTO gene, MC4R, AMY1. These are genes that you might want to take a look at because with the TCF7L2 variant, that can impair insulin secretion and glucose regulation. That can cause bigger postprandial after eating blood glucose spikes with normal fasting labs. And with the SLC38, that zinc, okay, transport, those variants affect insulin granular zinc handling and secretion, okay? That can alter the glucose sensor set point and the way that insulin responds. And when you have those dynamics, you can have higher A1C, more hepatic, the very low density lipoproteins, and small, dense LDLs. I mean, there are a lot of other things going on, that we could go through that can affect this. But when you're looking mechanistically, small, medium LDL, increase in numbers with normal LDL, um, uh, LDL number size, going back to the thing like the medium size. So your liver is overproducing triglyceride rich particles. They're not clearing the LDLs efficiently. Genes like ApoE, PCSK9, okay, you can find these in the cardiovascular profile, your reports. And when you're talking about insulin signaling variants, detox, oxidative stress patterns, that pushes the LDL toward these smaller, denser, stickier species. Okay. So, yeah, it really, I could totally nerd out on this, obviously. But it's really looking at the patterns, the genes, what are their lifestyle, the food, what is putting pressure on these genes to cause these things. But great question, great observation, that I find very fascinating. And I know that our time, and we had a few more questions. Is there anything that you wanted to say to wrap up for tonight's presentation?
Absolutely. Yeah, I just want to say thank you. I mean, I love getting to all these questions. I'm so sorry that we don't necessarily get to every single one, but we do our best and we do follow up. So thank you for being here, everybody. And just, I don't know that I really have a wrap up, but just that it's such a fun puzzle and this is such a key, right? The DNA 360 really was a key for me opening up and understanding what was going on for this person because sometimes it feels like I'm shooting in the dark as a practitioner if I don't have if I'm guessing and I'm not testing. And so this is what really helped me get there and gave me the confidence to say, okay, I know what I'm doing with this patient so that when she got tired or when she was exhausted or she was like, oh, I'm not getting better fast enough. I knew that I felt really confident to say, no, stick with it. This is going to be helpful. And she felt great at the end. So it's a really great tool to help me feel confident as a practitioner, I'm sure for others as well. So yeah, just thanks for being here, everybody.
Absolutely. And I strongly encourage anyone, if you have situations personally, if you are having any problems and you want to know more, you want to work with a practitioner who understands DNA and is able to interpret it in a functional manner. That is what we do. Please contact us. I know Vanny has dropped in the chat links. You can also email CEO at thednacompany.com, get some information. You can work with one of us. We can go over and look at different tests that you may need. Also, if you are a practitioner, we've got so many resources in our DNA university, in our student area. I also have office hours, and we are going to be doing these webinars twice a month starting next month. Well, except that we have a holiday coming up, but we are going to be expanding what we are doing so we can better address your questions and help to expand your education within the DNA University and your ability to use our tests to enhance your practice. So with that, thank you so much for joining us this evening. Dr. Crawford, wonderful presentation. Thank you so much.
Thank you. It was a pleasure.
Absolutely. We will see you next week at our next webinar on next Tuesday. All right. Take care, everyone.